Hello students, this is Geethanjali. In the last video, I told that the experiment performed by Avery, MacLeod and McCarthy did not convince the scientists that DNA was actually the genetic material. So another experiment performed by Alfred Hershey and Martha Chase proved to be an unequivocal proof that DNA was a genetic material. That means it was pretty much unambiguous and no questions were further asked. So they actually used a process called transduction. So what is transduction? So we know that bacteriophages are viruses that attack bacteria, right? So these bacteriophages attach themselves to the bacterial cell wall and they insert their genetic material into the cell and they back off. So after this, the bacteria treats the viral DNA or viral genetic material as its own genetic material, and it starts producing the materials that make up this virus, and the virus is formed. Okay, so it starts producing bacteriophages within itself. So at one point it produces so many bacteriophages that it bursts open. So and this process goes on. So knowing this, what Hershey and Chase did that was they took two cultures, okay? They grew bacteriophages here using radioactive sulfur and here radioactive phosphorus. So one thing they knew is that the viruses growing in this culture would have radioactive protein, and here they would have radioactive DNA, because we know that by then actually they knew that DNA had phosphorus in it and protein had sulfur in it, right? So we know amino acids that contain sulfur, right? Like cysteine and all. They just had to know what was actually that was being inserted into the bacteria. That is nothing but the genetic material. So this had three steps to it, okay? Infection, blending, and centrifugation. So infection means the bacteriophage inserting its genetic material into the bacteria. Blending in the sense the viral DNA and the bacterial DNA blend with each other. So basically that's what it means. Um, it starts producing more of the viral DNA because it replicates its own DNA as well as the viral DNA also, right? So later we take this infected bacterial cells and we perform centrifugation. So what happens is that these bacterial cells, they settle down and the viruses uh, with no genetic material in them, they form the supernatant. Okay, if in case protein would have been our genetic material, we should have found the radioactivity in the bacterial cells. But you can see here no radioactivity was actually found in these cells. So that means protein was not actually injected into the bacteria. So it is not the genetic material. In fact, the radioactivity was found in the supernatant. That means it was with the left out part of the viruses, right? So if actually the DNA was a genetic material, we should have found out the radioactivity in these cells, in the bacterial cells. So when actually the viruses containing their DNA as the radioactive material were injected, their radioactivity was also found in the bacterial cells. So this, with no doubts, proves that DNA is actually the genetic material.